Hi everyone, it's Mr. Sinti, and I'm happy today to be able to talk to you today about something called the chromosomal basis of inheritance. Let me start off by refreshing a little bit about the Austrian monk Gregor Mendel. Gregor Mendel died sadly in 1884, and here's a statue of him right here. And what's interesting is Gregor died in the monastery sort of in obscurity, meaning that no one really knew him, or his contributions weren't to the study of inheritance weren't widely accepted or recognized. And so it's kind of sad. It took biology, though, about 35 years to sort of catch up with what he was trying to say. It wasn't until like the 1900, in other words, the beginning or the dawn of the 20th century. We're in the 21st century now. So about 100 years ago, at around the turn of the century, biologists started to sort of get an idea of what Gregor Mendel was getting at. And I thought I'd include this little quote from Gregor Mendel a year before he died. I thought it's kind of sweet. And he goes on to say that even though he had some bitter times in his life, he was thankful that the pleasant and the good prevailed. That's kind of nice. And that his work in the garden and his scientific research brought him a great deal of satisfaction. And he was convinced that it wasn't going to be long till the whole world acknowledged it. And so, indeed, we have. And so, thanks to Gregor Mendel, we were able to move forward. Here's the question, though. Why did it take so long for biology to sort of catch up? It seems pretty clear what Mendel was trying to say about his patterns of heredity and his factors that he was talking about, and dominance and recessive. But here's the thing. What is a factor? You could talk about one factor dominating another in pea plants, and you can talk about how these factors assort randomly when there's two traits, but there wasn't anything physical. Like, what was Mendel actually talking about? What was it in the cell that was contributing to heredity? And so it took around the turn of the century in the 1900 for microscopy, or our microscopes, to sort of catch up. And so this is a picture of chromosomes under the microscope. And so biologists started to get a physical basis of the way chromosomes were behaving. In other words, we were getting a good idea of what mitosis and meiosis were all about. And as you may know from our previous discussions of inheritance, that's all about what Mendel was trying to get at. What he was trying to explain meiosis, although he didn't have the terminology or the microscope to do it. And so we're starting to see a pattern. In other words, when you could see here's a cell that's undergoing anaphase, you could start to predict that when you look at cells closely under the microscope, you could see that the chromosomes assort during anaphase. And it appeared to biologists that chromosomes were really important. They were able to measure the amount of chromosomes before and after, like diploid cells and haploid cells, and they were noticing that haploid cells, or pollen, had half the amount of chromosomes. And so it appeared to biologists at this time, about 100 years ago, that Mendel's factors were something called genes. In other words, segments along these chromosomes. So it appeared that these chromosomes, these physical structures that appeared in the nucleus of a cell, had everything to do with heredity. And so we know that today, obviously. We know even that the chromosome is made up of DNA. They didn't even know that 100 years ago. It wasn't until, do you recall, Hershey Chase and other scientists to determine that DNA was the heredity material. So we know that chromosomes come in diploid in pairs, and so like we could say like this one right here and this one are homologous pairs. And we know that if there's a gene there, it's the same gene that's there. They come in pairs. And so we know this to be the case. And so homologous chromosomes during meiosis separate. So if we had a cell like this and we had one chromosome there and one chromosome there, meiosis is all about separating the homologous chromosomes, and then meiosis 2 is all about separating the sister chromatids into 4 haploid cells. These are also haploid cells. And then those gametes, or reproductive cells, sperm and egg, fertilize each other and restore the diploid condition, as you can see here. So As I mentioned before, around the turn of the century, this sort of so-called chromosomal theory of inheritance began to take form. Instead of just Mendel saying that, round and yellow and green and wrinkled makes round and yellow, and then in the F2 there's 9331, it seems kind of magical. I mean, there's a prediction that of an outcome, and it's testable, but like, what is it literally? And so we're starting to get a sense that it's all about whatever these factors are, which we now know as genes. They're physically located along chromosomes, and these chromosomes physically separate during meiosis when pollen is formed, an egg is formed. And that is what is accounting for the outcomes that Gregor Mendel was giving us. And so that's a pretty big deal. I mean, we take it for granted, but it's a real important step in our understanding of heredity, because it goes from just sort of patterns to a physical basis, chromosomes. Okay, chromosomes. Well, let me introduce to you, in my opinion, what I think is one of the another great in the study of genetics, and this person's name is Thomas Hunt Morgan. He was an embryologist, really, who was studying at Columbia University in New York. And what's interesting is he contributed a lot to our understanding of heredity by sort of tracing a specific gene to a specific chromosome. And so he was able to really link a phenotype with a specific chromosome. And so that sort of began a huge discussion in biology. He was one of the first to associate this. And so, how does this story go? It's like you don't just sort of wake up in the morning deciding that you want to link a gene to a specific chromosome. And so this is a little bit of brief story of Thomas Hunt Morgan. As it turns out, that while working in Columbia in New York City, Columbia University, word arrived at New York from Europe around this time in the 1900s. And there was a botanist right over here by the name of, he was a sort of a Dutch biologist by the name of Hugo de Vries. And he was working in his experimental garden with these flowers called primrose. And he was breeding them, sort of like Mendel was. And as it turns out, when he was breeding them, one of the plants refused to breed. In other words, it just would not create any fruit with the other flowers. And so Hugo was quite taken aback by this. And so by our definition of what a species is, he believes that he had created or at least noticed a new species forming in his garden. It was a pretty big deal because around this time, just historically speaking, in England, Charles Darwin released his most famous book on the origin of species in 1859, and people were really wondering because though Charles Darwin laid out his idea of how evolution works through natural selection, he wasn't very good at explaining the genetics of variation. And so perhaps Hugo de Vries had stumbled upon something here. Maybe this is how new species come into being. You may be like, well, how do you mean they come into being? Well, Hugo, in his conclusion, wrote that he thought perhaps the reason that this plant doesn't reproduce with the other is that it had some kind of mutation. And so word was traveling across the Atlantic Ocean, and Thomas Hunt Morgan sort of got wind of this. And so he thought he would like to investigate mutations and see if he can discover how new species come into being. And so he wasn't specifically working on inheritance. And so, as it turns out, Morgan was a zoologist. He wasn't going to work with plants. And so he decided to work with this little tiny fruit fly called Drosophila, that's the genus, Melanogaster, which is the species. Drosophila means dung-loving. And so, as it turns out, it's an insightful choice for an experimental animal because it's very small and it grows rapidly. It's a fruit fly, but the truth is that it eats the little pieces of fungus that it's growing on the fruit. And one of the reasons that Morgan decided to work with this little tiny fruit fly is that, and this is a picture of Morgan here in his lab at Columbia, and he was growing these fruit flies in milk bottles, and all you have to do is give them some food, like down below here, you just give them some mashed up banana, and then mold starts to grow and the flies eat it. And so one of the things I just happen to have some fruit flies here. So if you could see them sort of flying around here. What's interesting about the fruit fly is, they're small and that you can have little containers. He didn't have a plastic container like this, he had glass. But you can grow hundreds and hundreds and hundreds of them. And they're really easy to work with. And so one of the things about them is that they're cheap, they're easy to find, and they're very prolific breeders. And so they're quick to reproduce. And as it turns out, not only are they quick to reproduce, is they have this really short generation time. So the male is over here. It's kind of small and has this dark abdomen here. And as it turns out, when it reproduces with the female lays an egg, and then that turns into a larva, and then that grows up. And in about a week and a half, this big larva crawls up out of the food, if you could see it here, and it creates this encasement called a pupa. And the pupa is where metamorphosis takes place, where the larva turns into an adult fly. And then the flies emerge. So the pupa gives rise to the female and the male flies. And as it turns out, this whole thing takes 2 weeks. And you're like, oh, that's great, but what's the big deal? Well, it's fast. And so if you were looking at heredity, if you're looking at a cross between males and females, you wouldn't have to wait very long. The P1 to the F1 to the F2, it's 2, 4. So in 1 month, you're already looking at F2 flies. And so it's really convenient. Not only is the generation time really short, as it turns out, when you look at the cells of fruit flies, and you were to pull one out like this, let me draw it over here on the bottom. If you pull a cell out like this, and you look at the nucleus under mitosis, it has 1, 2, 3, 4, 5, 6, 7, 8. It has only 8 chromosomes in the nucleus. And so that makes life a little bit easier to work with too, a lot easier than humans who have 46 chromosomes. So that was real convenient. And so Tom Morgan, he's working with the fruit flies. He's very excited. Well, I should say he's excited at first, but he keeps breeding them and breeding them and breeding them. And next thing you know, his whole lab is filled with these fruit flies, and he's looking at them, and he's looking for variants. He's trying to look for a mutation. And so as it turns out, he's looking for years, and he's getting kind of frustrated about it. And he's been breeding these flies, and he doesn't notice anything. And as it turns out, we were able to look back at his private journal, and it turns out that in letters to friends, he was saying he was just about going to give up, that he'd been looking at these fruit flies forever, it appeared, and he didn't see anything unusual, no mutations. And then finally, lo and behold, he discovered a male fruit fly that had a mutation and had white eyes, as you can see here. Now, I say white eyes is a mutation because all the flies have red eyes. I don't know if you could see it, it's really small, but all the flies have red eyes. When I say all of them, it's the most common. And so this is clearly some kind of mutation. So what we say in genetics is that sort of the normal character, like the most common, like for humans, for example, the most common phenotype for eye color is brown. The most common phenotype for hair is brown. And for fruit flies, the most common phenotype is red eye, and we call that the wild type, the most common. And so then we say, well, if red's the wild type, then the white must be the alternative or mutation. So it's some sort of mutant phenotype. So, it doesn't mean that it's a new species. It just means that the male had white eyes and the female has red. So no big deal. So what would you do if you're Morgan? Well, you'd probably, you'd want to cross these flies and see. You'd want to cross the male with the female and see what you get. And so, why would you do that? Well, you might want to consider if that mutation is dominant or if it's recessive. You'd want to investigate it. And so, of course, he did. And so when he crossed his very rare mutant white-eyed male fly with, of course, red-eyed female, all the flies were red-eye, it turned out that the F1, all of them had red eyes. And so, now Morgan was familiar with Mendel's work. He had been reading up on this, and so that was sort of what Mendel would have predicted, that the F1 would have exhibited the allele of one of the parents, and so it did. So all the flies were red-eye, and so he concluded that the red allele was dominant over the white, and he was correct about that. And it's like, okay, so now you got all these F1 flies and they're red-eye, and so what would you do with them? So you might want to cross them and see what happens. So you cross the red-eyed F1 flies, and if you recall, what would Mendel say if you crossed, like, let me just take you back for example. So if you had tall crossing with the short, like this, here's the Punnett square, you would get all talls, like that. There would be hybrid heterozygous. And then what would happen if you cross these F1s? You would get the classic 3 talls to 1 shorty, like that 3-to-1 ratio. So Morgan was kind of excited. So he's got all these red-eyed flies, and so let's cross them together to see what we get. Maybe we'll get the 3-to-1. Maybe we'll get it. And so he did. So he crossed the F1 flies, and he got the classic 3-to-1. So 75% of the flies were 75% of them were red and 25% were white. Or in other words, 3-to-1 phenotypic ratio. Now, if this was the only thing that Morgan did, we wouldn't be talking about him. So, I mean, it's very common in science for other scientists to repeat the work of others, but that doesn't necessarily mean they get all this recognition for it. So, as the story goes, I'm sort of making up the story slightly. As the story goes, when Morgan and his colleagues and graduate students in his fly lab at Columbia were counting the flies, 1, 2, 3, 1, 1, 2, 3, 1, 1, 2, 3, they were noticing the white eyes were kind of cool looking. Look at that. It's an interesting thing. They noticed that when they were counting the 3-to-1, there's a lot of flies here, so we're counting. When I say counting, you need to put the flies to sleep, or else, when I say sleep, I don't mean to kill them either. You anesthetize them. So it'd be hard counting them when they were flying around like that, because they'd get out. So you got to put them to sleep, and then when you're counting them with like a paintbrush, you're pushing them around like that. It's like, what did he notice that was a little unusual? Well, surprisingly, however, as it turned out, the wide-eyed trait, the wide-eyed color, appeared only in the male flies. Ooh. Now, that wasn't something that Mendel talked about. Now, you wouldn't expect Mendel to talk about it because he was working with pea plants, but I'm just saying that something was really, really weird, because out of these hundreds and hundreds and hundreds of flies, in this particular cross, he didn't notice any of the females with wide eyes. Now, you can get a wide-eyed female, we'll talk about that, but none of these flies, these F2 flies, were female that have wide eyes, so only males. And so this is a picture of Morgan. Look at him, he's pondering this. It's kind of a cool thing when you discover some sort of discrepancy through observation, and this is what gets the mind racing. This is the cool thing about science. This is why inquiry. I mean, he's dying. Like, he's wondering, why is it that males have the white eye and the females do not? There's got to be something. And it's like, no one knows, and so it's up to him to find out. What's he gonna do? Well, I'm not sure. It's like he concludes that, wouldn't you, that somehow the eye color was sort of linked to the gender of the fly, or the sex of the fly. In other words, somehow eye color had something to do with maleness. And you're like, well, yeah, but what does that mean, maleness? This is what he's thinking. And so, again, here's a picture of a fly. I know you're not so familiar with the fruit flies, but these are actually 2 males. This is one with white eye, and this is one with red eye. And it's like, well, I don't know. I don't know. Why would the white eye be present in the male more than in the female? I don't know where I would begin with this. Well, if you ask yourself this question, well, what is the difference between males and females? You're like, well, beside the obvious, the female's a little bit bigger, male's a little smaller, male's got this black butt here. As it turns out, Morgan figured that maybe there might be something on the cell level, on the cellular level, that separated males and females other than just the reproductive organs. So what he did was he took like a really thin pin or a little bit of like a toothpick, and he went in and he scraped out, sort of like the cheek, he scraped out some cells. But he didn't scrape it out of the cheek. He scraped it out of the fly's salivary glands right on the side of its mouth. He pulled out some cells, and then he looked at those under the microscope. So you check this out. It's pretty cool. So you're looking at, here's the female. You're looking at a female cell, and then you're going to look at a male cell. Now, this is a good question. Like, if I were to put one of my cells under the microscope, and you put one of your cells under the microscope, so female, male, would you be able to tell the difference between the cells? I mean, I don't think so. I think the cells look like this normally. Like, here's a nucleus. You're like, well, that doesn't tell me anything. Males and female cells sort of look like this, like an egg with a yolk. But here's the thing. This is a cell during interphase. What would the cell look like if it was undergoing mitosis? If it was undergoing mitosis, you would see chromosomes, right? And so what are chromosomes? So as it turns out, when Morgan looked at the female fly, he noticed like this. He noticed that there was 1, 2, 3, 4, 5, 6, 7, 8 chromosomes. And he's like, all right, there's a female, and then here's the male. 1, 2, same as the female. 3, 4, same as the female. 5, 6, same as the female. 7, same as the female. Ooh. The 8th chromosome right here was small. That was different in the male than in the female. And so as it turns out, let me go over here and circle this with the yellow. As it turns out, he noticed, this is a homologous pair, homologous pair, homologous pair. He wrote down that in females have 1, 2, 3, 4 sets of homologous chromosomes. Males, however, have 1, 2, 3 sets of homologous chromosome, and the last 2 are not homologous. And this one sort of looked a little weird. It was small, and so he called this, you may know this story somewhere in the past, he called these chromosomes in the female, both here in male, sex chromosomes. Write that, sex chromosomes. Because he thought they had something to do with, I'm not sure I can make that come back. Sorry. He thought that had something to do with how, well, here we go. All right, now I lost, sorry you guys. Apologize for that. Let's go here. Here we go, sorry. He thought that had something to do with gender, so he called them sex chromosomes. And so let me just do that again. 1, 2, 3, 4, 5, 6, 7, 8. And then 1, 2, 3, 4, 5, 6, 7, and then 8. So he thought these 2 had something to do with gender or sex, so he called them sex chromosomes. So what does that have to do with anything? Well, these ones, just for the sake of definition of things, these chromosomes right here, he kind of figured that those had to do with maybe the traits for wing and for legs or antenna and all this other stuff of a fruit fly. He called these other chromosomes autosomes. Auto means self, so autosomes, meaning like all the traits that we've been discussing so far have been autosomal. And so these ones are called the sex chromosomes. And so there was a difference between females and males. Females had XX and a male had an X and a Y. And you're like, well, again, does that explain why the males had white eyes? It might, because he was figuring, if it was anything, if the gene was located anywhere, where would you guess it to be? Since it seemed to show up unusually more in males, maybe it was on these sex chromosomes. And he kind of figured, females have eye color and males have eye color. Maybe the gene was located on the X chromosome. And if that's the case, for eye color, if it's located on the X chromosome, the female has 2 copies. That's nothing new. But the male has only 1 copy. Ooh, that's new. Let me repeat it. The male has only 1 copy for a gene located on the X chromosome because males only have 1 X chromosome. So what could this all mean? Well, what can all this mean is this. Check this out. This could mean that, let's go up here. Let's check this out. Let's go, let's not go there. Let's go here. Let's go like that. And let's say for example, we're talking about the fact that the female, whoops, sorry guys, that the female has an XX like this. This is a female, and this is the male XY. So I'm not drawing in the other chromosomes, I'm just drawing in the sex chromosomes. So what I'm saying is that Morgan realized that maybe the gene for eye color in fruit flies was located on the X chromosome, and what he called sex linked to the gender of the fly. And I made the X kind of large because it's located on the X chromosome. And you're like, well, what of it? Well, let's go back and sort of revisit his original cross. Do you remember he had, for example, this female that had red eyes? Remember that? Like this? Let me do superscript, because this is where the trait is. Like that. Red eye. This is the red eye female. And what are we going to do? We're going to cross it with a wide-eyed male. Do you remember we had that wide-eyed male, that mutant? Well, he didn't know it at the time, but as it turns out, the wide-eye male, like this, is made up of an X and a Y. And there's nothing on the Y chromosome. There's no gene. And so as it turns out, let me go little r for white, because he knew that the white allele was recessive to the red. So isn't this kind of unusual? So this is the cross. This is the parental cross. Do you remember what the F1 flies were? They were all red, and he didn't think anything was up with that. So check this out. Well, this is a diploid female fruit fly. And so let me just circle it here. So in order to make eggs from this, meiosis, you need to do this. And so what you do is you put an X like this, and here's our Punnett square. And then here's the male, X and a Y. So that means that the male, when the male produces sperm, it separates the X and Y chromosome like that and like this. So there's 2 kinds of sperm. There's an X sperm and a Y sperm. So as it turns out, check this out. This is white, this is red and red. So in Mendel's cross, what did he get? He got a girl with red eyes, heterozygous, a girl with red eyes, and he got a boy with red eyes and a boy with red eyes. Notice how the boy inherited the red eye from the female. In order to be a boy fly, you have to inherit the Y chromosome from your father. So all the flies were what? Red. He didn't suspect anything. But then check this out. When you go for the F2 cross, he took these heterozygous females right there, and let me just put it down here. Heterozygous females, so big R, little r, and he crossed it with the red-eyed male like this. And what did you get? You got a girl with red eyes, a girl with red eyes, a boy with red eyes, and boys with white eye. Oh, so here it is. Here's the 25%. I mentioned how it was 3-to-1 red to white, but only the boys had the white. Why do only the boys have the white? Because the boy fruit fly inherited it from the female. How about that? Isn't that interesting? So that explained what was going on. So that's pretty cool. Check this out. So let's go back up here. Maybe we will, maybe we won't. I think we will. So fruit flies have 3 pairs of autosomes and a pair of sex chromosomes, as we said. So they have a total of 8 chromosomes. The last 2 in a female are XX. The last 2 in a male are XY. And so this is a really awesome scanning electron micrograph of an X chromosome. And so Morgan deduced that the gene, very impressive, for eye color in Drosophila melanogaster was located on the X chromosome alone. And so he called it X-linked. And so as it turns out, the gene, just go right here, for eye color was located right there. And so you could see here, this is the cross that I was just working with. Here's the girl and the boy. Notice how white is, there's only one allele in the boy, and we're using a different type of nomenclature here. We're W+, which means red. So wild type is in the superscript plus. But as you can see, the F1 only produces red, and then in the F2, the white appears. Pretty neat. And so that explains, at least it does for mammals. You know, I don't want to completely simplify things, but like in other organisms, like in some cases, some reptiles, the basis of gender, how gender is determined, isn't always genetic. It can actually be temperature. So kind of some weird things. But as far as humans and other mammals are concerned, the chromosomes determine sex. In other words, if I'll just use myself as an example, since I'm male, I have 46 chromosomes. This one's my number 45, and this is my 46. My very last chromosome is a Y chromosome. It's a really small one right there. And so this determines gender. Now female, if you're female, you're XX. So your 45 and 46 are that. And so when you look at a karyotype like this, check it out, you're like, da, 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 da. These are the autosomes. Let me circle it to make sure. These are the autosomes right here. And you're like, huh, this last two right here, 45 and 46, are not homologous. This is the sex chromosomes. You're like, what am I? What am I? I am a male, if they're XY. If you're a female, you would look like this. You would be X and X. That's the karyotype of a female. You know what's cool though? This means that, you know, during meiosis, when you're making your eggs, you know how the homologous pairs separate, like this, separate, 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 which means your eggs will have one will have an X in it, and the other one will have an X in it. But watch this. If you're a male like this karyotype, and you're making sperm, so here's your diploid cell, and you're going to make some sperm. One sperm, now I'm not going to draw in all the others, but one sperm is going to get the X, and the other one's going to get the Y. Ooh, that's kind of interesting. So males determine gender. So this is a close-up of the X chromosome and the Y chromosome. So you following that? That's kind of neat. That was not known for a long time. You know, you can historically, this is kind of interesting when you talk about like royal families or whatever, like the king who wanted sons in order to inherit the throne and follow in the legacy of the king. The king always wants boys to carry on after him. As it turns out, if he's married to the queen and they're having, you know, girls and another girl and another girl, the king would sometimes become infuriated, like, my wife cannot produce the proper son, the heir to the throne. I must get rid of her. You know, I must get a new wife in order to have a son. When in fact, it's the king that determines the gender, right? I mean, here's the X and Y diploid of the king. So this is an X, this is the Y, like that. And so when you do the Punnett square, like this, here's the queen over here. That's why half the children in the world are female and half the children are male. It's 50-50. So how do you like that? And so, as it turns out, the egg produces only X chromosomes, and half the sperm have an X and a Y. So that's kind of neat. So it's about 50-50 chance of producing males and females. So sometimes people get confused with statistics, to say the least. They'll say that, you know, if a couple is like having 4 children and they had like a girl and a girl and a girl, 3 girls, and like somebody will say, well, you're due to have a boy. The next one's going to be a boy for sure. You've already had 3 girls. Now, the chance has no memory. it's always 50-50. You're never due for anything. It's always 50-50. Let's keep that in mind. So a little bit of a tangent here about gender because it's kind of interesting. But did you know that a baby, but at this stage, this is not a baby, this is considered to be an embryo, during the first 2 months of pregnancy. You ready for this? During the first 2 months of everyone's life, everyone was destined to be a female. And then in the second month, as it turns out, when you're 2 months old, that's when you start to see an emergence of whether or not you're going to be a girl or a boy. Now, what is that all about? And you're like, well, I thought you were saying something about like X chromosomes and Y chromosomes. So if this is the X chromosome and the Y chromosome, and say that this is a boy, and you're like, well, how do we know it's going to be a boy? Well, as it turns out, it has something to do with this Y chromosome, don't you think? And so as it turns out, it was discovered, I think fairly recently in the 1980s, that there's one gene in particular called the SRY gene that plays a really important role in determining gender. And so that's kind of neat. This gene has to do with the production of testosterone, and when there's testosterone, it turns the female plant into the male plant. So in other words, ovaries become testes, fallopian tubes become vas deferens, and as it turns out, that's kind of cool. And so we've determined it. It's located right here on the sort of the short arm of the Y chromosome, the sex determining region, or SRY gene on a male. That's kind of neat. And so, as it turns out, sex linkage, in other words, the genes associated with the X chromosome, this pattern of heredity isn't just in fruit flies. This could be applied to humans as well. And so they give us unique patterns of heredity. And so in addition to determining gender, this sort of the X chromosome has several genes on it. It has many things. So let's look at those real briefly. So what we call this type of inheritance sex-linked. And so like in the transition from the fruit fly is that the thing to get from this is that the male flies showed wide eye more commonly than the female. And so as the reason that is the case is that when you see there's only one allele on the male. So the male over here, if you will, this is simplified Punnett squares. Males are kind of like whatever you see is what you have or what you are. Like in other words, if you have little a allele, then you're going to show it. So in other words, if you have little r for fruit fly, you're going to be white. And if you have a big A or a big R, you're going to be red. And so males are either white or red, so they are what they are. But girls can be, since they're two traits, since there's homologous chromosomes, could be homozygous, they could be heterozygous or homozygous recessive. And so there's nothing new in terms of sex linkage for a female, but for a male this is different. And so here's a picture of the prince. Talk about royal families. Do you notice here the prince is looking a little lacking in the hair? We call this baldness. But sometimes it's called male baldness. And you're like, huh. Now why is that? Because you know this. It was just looking around in society, it seems like males are predominantly the ones going bald. Now why is that? Why is this the case? And you're like, I don't know. It's probably a male curse of some kind. Well, no, it has to do with the fact that, you guessed it, baldness. Let's go recessive. Let's say that baldness is located on the X chromosome. So if you're this, you're bald. And if you're this, and you're a male, you have normal hair or thick hair. But here's the story. Like, if you're a female, these are your possibilities. If you're female, you could be normal hair, you could be heterozygous for hair, and you could have the bald allele, but you're not bald. Or you could be, women can be bald, but it's more rare because you have this other allele backing you up. So you can be a carrier for baldness and still not be bald, whereas males are either going to be bald or not bald. Something like that. And so, just to point this out, the chance of a female inheriting a double dose of the mutant allele is less. In other words, for a female to be bald, your father has to be bald and your mother has to be a carrier for baldness. And so the chances of that are a little bit less likely. And so, I mentioned baldness, but, there are some other human disorders that are sex-linked that are far more serious than baldness. And one of them is muscular dystrophy. And that affects 1 in every 3,500 males born in the United States. That's fairly common. The numbers for female is that it's very, very, very rare. You wouldn't hardly find any females with this. Now, why is that? Because as it turns out, this muscular dystrophy affects the muscles, the neuromuscular junction. And individuals, it's very sad, these boys don't live very long. And they can be very weakened and spend their childhood in wheelchairs. And so as it turns out, if this little guy right here has muscular dystrophy, it's really sad to even talk about it. But if he has muscular dystrophy, what's his genotype? His genotype is XY, and muscular dystrophy, let's go little m for muscular dystrophy. So that means that he has it. So in order for a female to have muscular dystrophy, the mom has to be a carrier of muscular dystrophy, and the boy has to have muscular dystrophy. And as it turns out, it's just very rare for somebody who has muscular dystrophy to have a child, let alone with a woman who's a carrier for it. And so here's how you can get a female who has muscular dystrophy. Do you see how that's really rare? And then even still, the female could be a carrier, and then they could even produce a male over here that's normal. So it's kind of fascinating. And so another common example is, you may have heard of this before, it's a disorder called hemophilia, blood loving. It means that the person has a very difficult time having their blood clot. And so as it turns out, this is far, far more common in male boys than in females. And you're like, why is that? It used to be called the royal disease because it was found in the royal family a lot. And as it turns out, that because the royal family was reproducing with cousins and sort of keeping everything close to home, it showed up very, very commonly. And so as it turns out, in order to, let's say that a female, or let's say a male, a normal male would be this, or I'm sorry, a normal female would be this. Here's a female who's a carrier for hemophilia, and here's a female who actually has hemophilia. Now, it's very rare to get that female, whereas you can get a male with hemophilia. And you're like, how do you get that? Well, the mom could have normal blood, she's a carrier, and the father is normal. You're like, well, wait a minute. If the father's normal and the mother's normal, how do you get anyone with hemophilia? As it turns out, like this. The male can have hemophilia, and the male boy inherits hemophilia from the mother, because it's in order to be a male, you get the Y from the father and the X from the mother. And so it's a 25% chance, but it's very rare, as I was saying, to be a female hemophilia, because the father has to have hemophilia, and that's rough, and the mother has to be a carrier at least. And so here's the last example that I want to talk about that's sex-linked. Take a look at this picture right here. Do you see anything here? This is something called red-green color blindness. It's a particular type of color blindness that is very common in males. And if it's very common in males, you're like, ha, it must be sex-linked. And then, the gene for red-green color blindness is on the X chromosome. And males show it more because they have only one copy of the allele. But can you see this? This is a number 5. If you can see this, if you can't see it, then maybe you're colorblind. What's that? 7, like our quarterback for the 49er, Kaepernick. What's this number? Wait a minute. It's not a number. It's a sailboat. It's fooling you there. You're like, what's this number? I don't know if you can tell. Scary, huh? So watch this. Like when you're doing these kinds of when you're doing these kinds of studies, check this out. Let's do this. We'll close with this example. Let's say that we're going to study red-green color blindness, and you knew someone in your family that had color blindness, and you're like, gee, I wonder if I'm going to get it. You're like, well, you're not going to get it because if you're not colorblind, it's not a disease that you get. It's not a pathogen. So the question that you're saying is, like, well, I knew someone in my family. I'm wondering if my children or my children's children will have this. How will I know? How will I know? And it's like, well, I need to study the family tree. And so the way we study family trees in genetics is this. We can say that a male, let's draw a male as being a square or a box. And the way we do this is like that, and we say that the line going across to the female means that they're married. So here's a couple, it's a boy and a girl, and they're married. And then the line coming down means that they're going to have some children. And so let's say that this couple right here has 2 children. And let's say that they have the first child. This is the parental generation right here. This is the F1. Say they have a boy and another boy. And you're like, ooh, this boy, let me shade it in like this. Let me say that the shaded in square means that this boy, let's go with the red-green, it has color blindness. And then we'll go, this first boy marries this woman right over here, and they have children. And they have a girl and a boy, like that. And I'll say this boy is colorblind. And then let me go over here and say that, okay, this guy is married to this girl, whose parents up here were obviously a boy and a girl. And then this couple right over here decide to have 2 children as well. You may have done this somewhere in your past. This is called a pedigree. You heard that before, a pedigree. So this is the parental generation or grandparents, parents, and then kids. And I could make this up. I can say, that the, and let's just do this. I'll use a different color and go, okay, like what's the genotype of this guy or grandpa? Like, what's the genotype of grandpa? And you're like, I don't know. I guess he's XY. And what's grandma? I guess grandma's XX. And you're like, well, if it's not shaded in, that means that grandpa does not have color blindness. So color blindness is recessive. So I'm going to give him a capital C. So he's normal. Now, I don't know what grandma is, but the story is that the first son was normal vision. So if he's normal vision, this guy right here is normal vision, he must be big C, XY big C. You're like, well, what's this guy? Well, this guy is XY since he's a boy and he has a little c. Look, he's colorblind. He's colorblind. You're like, well, where did he get that little c from? He must have gotten it from his mom, right up here, grandma. And so as it turns out, grandma's got normal vision. What's going on with that? So as it turns out, grandma must be, yes, heterozygous. So she's a carrier. She's a carrier for color blindness. We didn't even know it. So then this boy is colorblind, and so he's thinking, I better not have any color blindness in my family. And so I'm going to make sure I marry this girl over here. This girl doesn't have any. He makes sure that she takes that color blindness test, and so she's apparently normal. But we don't know if she's what? Homozygous or heterozygous. But then he looks into it and says, well, her parents are normal vision. She's probably fine. But as it turns out, they have a son who is normal vision, and so that's great. But then they have a daughter who's colorblind. He's like, oh no, colorblind daughter, what's going on? Well, in order to have a colorblind daughter, where did she get that colorblindness? One from the father and one from the mother. See that? And so, therefore, this is normal. And then what's this woman right here? Can we tell? Yeah, she also has to be a what? She has to be a carrier for this one to take place. Now over here, what's this? Here's a female who is normal. So she's got to have one big C. This guy, so they have 2 normal parents. They have a normal child, normal daughter. But then, a colorblind son. So colorblind son. But interesting, where did the color blindness come from? And it came from the mother. So this is kind of curious. Can you even tell what this girl is? And you're like, well, she's normal. Check it out. She could either be pure normal, in other words, homozygous normal, or, I'm going to put an or down here, or she could be a carrier. It's hard to tell just from looking at it phenotypically. So, hope you enjoyed our conversation today about chromosomal basis of inheritance, and then we segued into sex linkage and traits and some various disorders. And I hope you're not seeing too much red. So thanks for watching.